It gives me great pleasure this morning to introduce our speaker, Laura Itzhaki from Cambridge University. She's head of the Department of Pharmacology and co-director of the Cambridge Academic of Therapeutic Sciences. And Laura's work is very much targeted to biotherapeutics and using the cell's protein handling technologies to advance our medicinal technologies. And with that, I introduce Laura to tell us about harnessing the cell's protein degradation machinery, nature's way.
Great, thanks very much for the introduction, and thank you to the organisers for inviting me. So, before I get on to the science, I'd like to do a couple of shout-outs. The first is to a BPS meeting we're organising in Cambridge next year in April. Please do have a look at it. We've got a great lineup of speakers, and we very much look forward to seeing you there. The second shout-out is to a new scheme that we set up in Cambridge. So the aim of this scheme is to mitigate the effect of leave on researchers. We're talking maternity leave, but not exclusively maternity leave, so it could be parental leave, it could be sick leave. And the way we're doing that is by providing research support to people. So this is how our pilot is working. These are the aims, as I say, to mitigate the impact of long-term leave, such as maternity leave. Also to address the challenges of short-term replacements for people on leave. So, for example, even if you have the opportunity to get extra funding for to cover somebody when they're on leave, it's very hard to recruit somebody for, say, 6 months. It enables the research to continue without a break, and it allows us to sort of make our research community more equal and inclusive. And lastly, it reduces the bias in recruitment, particularly of young women of childbearing age. So one of the main aims of setting this up was, as I said, to address gender imbalance in the academic pipeline. And you can see that here. So this is from a Cambridge Equality and Diversity report from a couple of years ago. And you can see here, actually, we're looking at academic staff and different grades. You can see that the number of male professors is almost the same as the total number of female academics in Cambridge. So still huge disparities in the academic pipeline. And we're kind of recognising the fact that in many other areas, including professional staff in our institutions, it's business critical to have maternity cover, for example, but researchers just don't get that. And also it's kind of acknowledging that really women do shoulder most of the caring responsibilities, and this doesn't just simply mean childcare, this can mean caring for elderly relatives as well. So, as I said, we've set up this pilot scheme in Cambridge, but please do consider setting up a similar scheme in your institutions. We can provide you with all the material you might need, so case for support, examples of adverts, researcher application forms, reporting and impact. So please do either get in touch with me or via this email address here. And this is a really nice article that came out in Nature earlier in this year on roving researchers, which are schemes that there are worldwide, actually. So this isn't just Cambridge. We weren't the first to do it by any means. Okay, so on to my science now. So a major focus of our research in recent years have been these proteins here, tandem repeat proteins. So these are a class of proteins composed of small structural motifs, you can see on the top, repeated multiple times in tandem to produce these very beautiful circular, superhelical and elongated structures. And a key feature of these proteins is that all the interactions are short range. So all the interactions are either within a repeat or between adjacent repeats. And I'll come on to why this is really important next. So this very simple linear structure and the absence of those sequence distance contacts that characterize globular proteins enables us to redesign these proteins in a very simple manner by taking an approach of design and repeat. We start with what are called consensus-designed artificial proteins. So, and I'll come on to what they are next. These are very, very stable, and so we can produce them recombinantly in very high yields. And this is an example of their stability. So here we're looking at anchorin repeat proteins. This is thermal unfolding, so percentage folded protein, not unfolded protein as a function of temperature. Actually, sorry, it's not percentage. This is a circular dichroism which measures secondary structure content. And these are two natural anchorin repeat proteins. You can see that they have very modest thermal stabilities around P16 is particularly unstable. It has a melting temperature around body temperature, and gankerin slightly higher. And these are a series of consensus-designed anchorin repeat proteins. So by consensus design, what I mean is we take a family of proteins, for example, anchorin repeats, we align them, we take all the conserved residues, and we make proteins comprising identical conserved residues. And these have been found to be conserved for a reason of stability, and that means that when we make these consensus-designed proteins, they're extremely stable. And so you can see, even for the smallest one, which is just 3 repeats, I have different ones here from 3 to 13 repeats, even for the 3 repeat protein, which is around 100 residues, it doesn't unfold till beyond 80 degrees. So very, very stable proteins. And that's important for our design approach. This very stable scaffold can tolerate what we call grafting of short linear motifs, binding motifs known as SLIMs. We can graft them, as shown here on the right, either onto alpha helices in green or onto loops between repeats. And the modularity means that the repeats are self-compatible. There's no constraint on numbers of repeats. We can design them independently and then put them together complementarily in any order. And we can also design them to display multiple functions in very precise and predictable configurations. So that's shown here. We can take one with a grafted alpha helix and one with a different function with a grafted binding peptide on a loop and make a multifunctional protein. We've used these artificial binding proteins as inhibitors, and some examples are shown here. So on the left is, we took a peptide from the protein NERV2, we grafted it onto this loop between adjacent repeats, and this is a potent inhibitor of this protein here, KEAP1, which is an important target across multiple diseases. The middle and the right example are repeat proteins where we've grafted an alpha helix onto a terminal alpha helix of the scaffold. So this is RAS. This is an alpha helix from one of its binding partners, SOS1. We've taken the key interacting residues and grafted it onto this helix to make a RAS binding protein. And in this case, this is MDM2. We've taken a peptide from the protein P53, grafted it onto this helix of the scaffold to make an MDM2 binding protein. And these binding affinities shown here nicely recapitulate the binding affinities of the proteins from which they were derived. And they're much higher binding affinities than the isolated peptides, which aren't constrained. So for example, if we just took this alpha helix here, if we took the residues on their own as an isolated peptide, they wouldn't form a helix and they wouldn't bind very tightly. So, as I say, because these are modular scaffolds, we can have multivalent and multifunctional display. And we can do this by grafting multiple of these binding peptides on in lots of different ways. Some of them are shown here. So, for example, we can linearly repeat the binding motif. We've drawn it as a linear array, but actually many of these repeat proteins form a superhelical structure. So you can see the binding loops in red are displayed away from each other by in this case by 90 degrees when we put them on alternative loops. We can also display them in different ways by fusing them to oligomerization domains. So this is a trimeric domain that forms trimers. And then we have a very different way of displaying these binding functions, and we can display many binding functions within one protein. And multivalency, we think, is very important, particularly for multivalent targets, where single valency inhibitors often don't work very well, but also for monovalent targets where we can enhance the affinity and the specificity by avidity effects. So this is exactly what we've done. This is grafting of one or more copies of binding peptides for the proteins tankerase 1 and tankerase 2. These proteins upregulate Wnt signaling in various cancers. They do this, so tankerase paralates axin, and then axin is then targeted for degradation. It's unable to form this degradation complex with beta-catenin, which is then upregulated. So we've made either single valency binding proteins for tankerase or multivalent binding proteins using either this linear display or this trimerization domain. And we've shown that because tankerase itself is multivalent, so it has multiple binding sites for this binding peptide, our binding proteins sequester tankerase into these very large higher order assemblies in the cell. And we think that that process of sequestering tankerase is what makes these inhibitors particularly potent. So they are the single valency proteins are less potent than the multivalent ones. Here we are delivering the proteins encapsulated in lipid nanoparticles, and you can see that they have reasonably high potency and also much higher potency than existing small molecule inhibitors of the catalytic domain of tankerase, shown here in green. So these inhibitors work quite well prophylactically, but to be clinically useful, they need to work interventionally, and they're not very good interventionally. They're not good if Wnt signaling is already high, but our multivalent binding proteins have much higher potency in an interventional context. So we deliver these proteins into the cell, as I say, using lipid nanoparticles, but we wanted to see if we could engineer the proteins themselves to be cell penetrating. And this is exactly what our collaborator, Itzhaki Cortarena, has done. She's in San Sebastian, Spain. So what she does is she takes these same repeat proteins and engineers metal coordination sites into them using cysteine residues or histidine residues. Using these proteins and a very simple assembly technique, these proteins can be formed into nanoparticles. The nanoparticles are small, up to around 5 nanometers in size. They're very stable. They can be stored for a long time, particularly in the fridge. They're photoluminescent, so their main use for them has been as looking at imaging and theranostics. But importantly for us, metal coordination enables these repeat proteins to enter into the cell. Also, I should say they're biocompatible, they're non-toxic, and they're very photostable. So we've designed these metal nanoclusters. We've put in histidines into our repeat proteins to coordinate iron. So these are protein-stabilized metal iron oxide nanoclusters. And here, we're looking at these binding proteins that we've designed by grafting this short peptide from NRF2 onto the repeat protein scaffold to inhibit KEAP1. And we made single valency and multivalency versions of these. And this is results in a lung cancer cell line. You can see that these metal protein-stabilized iron oxide nanoparticles can get into the cell. They bind to KEAP1, and they displace NRF2 and increase its levels and allow it to be transcriptionally active. So that here we're measuring the relative transcriptional activity of in the cells. And you can see that these are bare protein stabilized iron oxide nanoparticles. Here we have the single valency and the multivalent versions. The multivalent versions seem to be much more potent than the single valency equivalents. So having shown that we could engineer single functions into these proteins and make them multivalent as well, we were next interested to see if we could make them multi-specific and use them for induced proximity. And so we call these proteins, design proteins, polyproxins. And this was back in 2018. These are some of the people who were around then. Some of them have reappeared or really not gone away. But actually, we were inspired by work that we'd done in 2002, so a long time ago. And this was on a protein called CKS1. This is the protein here. It's a very small protein, but very mighty protein. So CKS1 is a natural proximity-inducing mini-protein. It packs binding interfaces for 3 different proteins. So those binding interfaces shown in red, blue, and green onto its surface. It uses 70% of its surface to do so. It's a very small protein, 79 residues. And this is how it works. So this in red is P27. It's a cell cycle inhibitor. Oops, where's my cell cycle inhibitor. It is ubiquitinated by this E3 ubiquitin ligase, SCF skip2. And then P27 is targeted for degradation by the proteasome. But that doesn't happen in the absence of CKS1. So what CKS1 does is acts as a proximity-inducing protein. It brings the E3 ligase into proximity with the target P27. CKS1 also binds to CDK2, which is bound to P27. So this combination of the 3 binding sites means that P27 is efficiently presented to the E3 ligase to allow processive ubiquitination to occur. And knocking out any one of these 3 binding sites abrogates that function. So this is what we're doing. We're aiming to make synthetic proximity-inducing proteins. This is a schematic showing the cell's quality control and degradation machineries. And it shows how diverse they are. And we understand these pathways very well. We know the different components. We know many of the structures for the different components as well. And there's a common underlying theme, and that's shown in red, in the red box here. So in all these pathways, the substrate is recruited to this machinery via a short peptide motif, so a SLIM again. So for example, in the case of ubiquitination, substrates contain what are known as degrons, short peptide motifs, that are recognized by E3 ubiquitin ligases. The E3 then ubiquitinates the substrate, and it's targeted for degradation in the proteasome. And these short linear motifs are common across these very different machineries. So what we're aiming to do with these peptides is use them as ligands to co-opt the machineries to drive degradation of disease-associated targets. So we're using those peptides to co-opt these machineries. We're also using peptides to recruit targets. So our polyproxins are bifunctional polypeptides that have a target binding peptide and a binding peptide for an adapter so that it can be taken to one of these machineries, either to the ubiquitination machinery or to the lysosome, or bypassing this ubiquitination machinery directly to the proteasome. And we hope by using peptide ligands that we might be able to firstly access more targets. So you'll all be familiar with PROTACs, proteolysis targeting chimeras. These are small molecules that have been generating huge excitement. But to date, they have been focused on targets that have already been drugged. So there still remain a huge number of targets that haven't been drugged by small molecules or antibodies. Also, many targets that appear to be resistant to kind of conventional targeted protein degradation approaches, for example, such as aggregates. We also hope to be able to access much more of the cell's degradation machinery using peptide ligands. So this is an example of one of the assays that we use. This is a cell sorting assay. We use it to get an idea of the degradability of our target. So we make stable cell lines with the target fused to GFP, so stable expressing these fusion proteins. We transfect with degraders, and so these degraders, as I said, have a target binding motif and a degradation motif. They're fused to an M-cherry with a cleavable tag. And then we use flow cytometry to sort the cells and determine the levels of degradation by the loss of GFP fluorescence. So we're looking for a shift to quadrant 1 here. And this is some data for one of our targets. This is BCL-XL. You can see that we can get reasonably good degradation using all of these different approaches, so either targeting to E3 ubiquitin ligases directly to the proteasome or targeting to the autophagy lysosome pathway. And back to this idea of multivalency, this is a really key feature of many of these degradation machineries. So one example is E3 ligases and their substrates. These tend to be multivalent. So, and there's a number of ways that this is achieved, and these are shown schematically here. So for example, the substrate often has multiple degrons that can be recognized by the E3, so that's shown in B, C, and D. But also the E3 has the capability of being multivalent. So, and this can occur, for example, by oligomerization of the E3. And this is a really interesting example. This is an E3 ligase spot shown in different shades of purple and pink here. It forms very large oligomers. A dimer of dimers is shown here for simplicity. So it's multivalent, and then its substrates like E3 tend to be intrinsically disordered and have multiple copies of spot binding motifs. So these are also multivalent. And the combination of these two multivalent proteins means that these can assemble into very high order cross-linked dynamic structures that enhance ubiquitination, enhance the processivity of ubiquitination, and allow proteins to be degraded much more rapidly. So for this work, which is a collaboration with Andy Wilson at Birmingham, Dec Wolfson at Bristol, and Tom Edwards at Larkin in the States, we've been using a different scaffold from the repeat proteins I talked about. This is it's also a repeating scaffold. This is a coiled coil scaffold. And we chose this scaffold because we believe it allows us to introduce multivalency in many different ways into the scaffold. So we can introduce multivalency by using the fact that coil coils form oligomers, so not just dimers that you might be familiar with, but these are de novo designed coil coils from Dec's group. They can be designed to form even higher order oligomers. But the other way of introducing multi-specificity is by decorating the two helices, say in a dimeric coil coil, with a target binding peptide and a degradation module. So that's what we've done. The first approach, where we're using the fact that these are oligomeric, is shown here. So we used a combination of a binding peptide for BCL-XL, different binding peptides for different components of the cell's degradation machinery, and then different types of coil coils. And we used parallel helices and anti-parallel helices. The binding peptides are decorated on the ends of these. So you can see for a parallel helix, parallel coil coil versus an anti-parallel coil coil, these binding peptides will be configured relative to each other in a completely different way. And you can see for some of these designs, we get very efficient degradation. This is looking at endogenous BCL-XL levels. We've also looked at grafting BCL-XL binders and E3 ubiquitin ligase binders onto the coil. So these are de novo designed coils, as shown here. So one helix binds to the E3, one binds to the target. And again, we get, for some of our designs, good levels of degradation. We've now extended our efforts to membrane proteins, in particular GPCR, so I feel like I can start to come to pharmacology conferences and feel like I belong, because there's still, of course, many GPCRs are very well drugged, but there's still a lot of space for improvement or for targeting those GPCRs that are not very well drugged. And there are multiple points in membrane protein and secreted protein life cycles that can be harnessed for degradation. So some of those are shown here. What's been done so far in this space is to piggyback on internalization receptors. So for example, LITACs, lysosome targeting chimeras. But we also think the scope to use the cell's internal machinery for degradation, so to capture proteins as soon as they're synthesized and before they reach the surface. So for example, targeting them to E3 ligases and the ER degradation machinery, which of course is very powerful at keeping damaged receptors, for example, under control. So we've looked at the GPCR CCR6. This has not been effectively drugged to date. On the left, this is degradation of CCR6 GFP fusion. You can see, again, using different machineries, ubiquitination, direct to the proteasome, or lysosome targeting, we're able to see good levels of degradation. And on the right, we're looking at downstream signaling. And you can see that we have the degradation of the target is able to control signaling. So to finish, we're now looking at another approach which we hope will overcome some of the challenges of targeted protein degradation. We've called this targeted protein degradation MAX. And this is taking the concept of induced proximity a step further. We're using two different approaches. The first is shown here. So what we're doing here is we're designing bifunctional polypeptides that will induce liquid-liquid phase separation. And that, we hope, will bring effectors and targets into much closer proximity to really accelerate the rate of ubiquitination and degradation, for example. And the other approach we're using is nanoparticles. So we're by condensing targets and, for example, E3 ubiquitin ligases on the surface of a nanoparticle using targeting ligands for both the E3 and the target. We hope, again, to accelerate the degradation mechanisms and overcome some of the challenges, for example, hook effects, and the requirement for a very specific orientation of the target onto the E3 by really sort of condensing the target and the degradation machinery on this surface. So I'd like to finish by thanking my colleagues who have done all this work, in particular those shown in red, and my collaborators both in Cambridge and elsewhere, and funders. And thanks very much for listening.
I should mention at this point that you have the capacity to upload your questions using the app, the program app, and the QR code. So feel free to do that. And with that, anybody that's got any questions, you can stand up or load. Laura, to kick off, bioavailability. There must be some challenges that you're facing there.
Yeah, actually, I didn't mention it. I didn't mean to mention it. Yes, so of course, we're saying that there are still lots of challenges with using small molecule degraders such as PROTACs, and these are around being able to access targets that don't have small molecule ligands, but also access many of the hundreds of E3 ubiquitin ligases, for example, that don't have small molecule ligands. But of course, the challenge for biotherapeutics is getting them into the cell. But we have been exploring in various collaborations, including with AstraZeneca, using mRNA, lipid nanoparticle encapsulated mRNA to deliver these proteins into the cell. And that's the approach we will probably take.
Okay. Microphone 3.
Thanks, Laura. I was curious if it's possible to use, or if you've perhaps noticed, whether proteins like CKS1 are useful for identifying or predicting targets that are going to be particularly sensitive for degradation approaches. Because the questions always arise within the induced proximity field about whether you're enhancing existing interactions or you're creating novel interactions.
Yeah.
And you know, so CKS1 struck me as, you know, is this part of a larger family? Are there proteins out there that are already identifying targets in the cell that are going to be very susceptible for degradation?
I think that's a very good question. Just trying to think how to answer it. So there are kind of, CKS1 is an example of an adapter I mean, E3s have substrate recognition subunits, and CKS1 binds to one of those, so it kind of modifies or modulates the existing recognition of SCF skip2. And you know, people have speculated that it has this requirement for 3 binding sites. P27 has this requirement that it needs CKS1 because it needs to be tightly controlled. In terms of looking at what's already out there, I think it is useful, for example, to, when you're thinking about designing molecular glues, where it's really challenging, or it could be really challenging to bring a new E3 to a target, 2 proteins that don't naturally interact by a single small molecule that kind of binds to both parts, both interfaces on the protein, simultaneously, to develop molecular glues to re-glue things back together. So for example, we're looking at SPOT, which is mutated in lots of cancers, and one of its targets, AR, which is also mutated in lots of cancers, and looking at those mutants and trying to design molecular glues to re-glue them together. So rather than bringing a target to a new E3 ligase, to use the natural binding partners, the natural E3 ligase for that target. But I don't think people have looked specifically at whether some proteins are more sensitive than others. It's an interesting question. Yeah, it probably doesn't answer your question, but I can think more about it.
Thanks for the opinion. No, thank you.
We've got an online question from Emma. What sort of disease areas do you think this sort of approach would be most suited for?
So we're looking at targets that don't have small molecule ligands, and those have been cancer targets, for example. But there's also many receptors that still lack ligands, and people are using small molecule approaches, but they're actually quite complicated. They're often small molecules that are conjugated to antibodies, for example. And ours are purely genetically encoded. So we think any disease where the targets are known and there are no small molecule ligands, this could be an easier way of addressing that target. But also, generally for targeted protein degradation, it will be really important for targets where inhibition of a single function is not enough. So, for example, tankerase is a multi-domain protein. It has a catalytic domain, but inhibitors of that catalytic domain aren't always effective. It has some non-catalytic scaffold functions. If you knock down a protein, then you will remove all of those functions. And also, some of these small molecule inhibitors of the catalytic site of proteins, for example, are very generic. There are lots of off-target effects. So we're aiming for intracellular targets in cancer, but we're also looking at, for example, pain. There's a lot of targeted protein degradation is on cancer, so we're trying to look for other areas, for example, pain, where we're looking at receptor degradation. But we're encouraged because in the States there are a couple of startups that have come out of stealth mode doing exactly this on intracellular targets. So they, I think, encouraged by the mRNA vaccine for COVID, people are encouraged to look again at biotherapeutics and potential delivery strategies. And by the time we get our therapeutics working well, we hope that there will also be advances in delivery approaches.
Seems to me there's an opportunity for topical application, Laura, and maybe psoriasis.
Yeah, I mean, absolutely. We've only chosen cancer because it's kind of an obvious one, and we have some nice targets that we understand well, whose biology we understand well. But yeah, absolutely, there are other applications where biotherapeutics might be easier to deliver, both intracellular and extracellular. Yeah, absolutely.
Any more questions? I can't see anybody. Oh, there's one.